Hello everyone. If you like my video, please subscribe, like and share it for watching upcoming videos. Thank you. Hello students. I hope everyone is doing well. Today I am going to continue the same chapter, reproduction in plants. Today's topic I will discuss artificial vegetative propagation. So let us start. Artificial vegetative propagation. This method is being artificially done by farmers, horticulturists, and what are the main processes involved in this are cutting, layering, grafting and tissue cultures. These all four methods we are going to discuss in details now. First method is cutting. In this method, the stem is cut into small pieces. Means stems are cut into small pieces with each bearing an axillary bud. You have to underline this one. Each bearing a axillary bud. The cut ends are planted in moist soil. After a few days, they strike roots, grow into a new plant. This method is normally employed for propagating plants like what type of plants comes out. Last line also you underline which example come under this. Lemon, China rose, sugarcane, all and many are there are coming under this category in which we are cutting a small stem bearing an axillary bud and they are planted in a moist soil and after a few days we find that the root comes out and it gradually grows into a new plant. Now next one we will see, this is a very common one, cutting one. Now we will come to the layering. What is this layering says us? In this method a portion of one of the lowering branch. Can you see from this diagram here also? And from here also you can see the diagram that the picture is there, that lowering branch. Can you see these are the upper branches? This is the lowering branch. Lowering branch is made to bend down to the ground so that it touches the soil. And the ring of bark means you must have seen that the bark is there in the outside of the stem. So they are being removed first, removed from that portion which will go and touch the soil, which is then covered with soil. Then again it is inserted inside the soil and then covered it in with the soil. Some heavy object like a small piece of stone or a brick is kept on the branch. They are kept on the branch so that it will not come out from the soil because the tendency will be there to come up. After a few days you will find that the branch gives out root. Then what happened? It start giving the roots. You can see here it start giving the roots after in a due course. When it start giving the root, then what happened? It doesn't need now no more help from the parent plant. Then it is removed from here. It is being cut off from the parent. Then it is cut off from the main plant. It is continued to and it is now individual to grow independently. And this type of method propagation is used in the common plant. Can you see mint, rose, jasmine? You can take the example. These are taking this layering method. Now coming to the grafting. Grafting is also one of the method which are coming under this. Rose, mango, guava. You can see that rose is a very common example in which every method can be developed. So a small shoot or bud of a desired variety of plant is intimately fixed on the stem of other plant. That means small shoot or bud of a desired variety. Suppose I want to make this variety of plant to fix into the another variety of rose plant, this rose plant to the another variety of the rose plant, then what I will do the process grafting. Small shoot I will take which or bud of that because you know they bud bearing they develop into a small plant, fix on the stem of another plant of some related species. Related species also or same also. The plant receiving the bud, that means suppose this is the plant, you can see here also the plant which receives the bud, the stem which will come and get attached, the receiving one is known as stalk and the small shoot which is being attached to there is known as the scion. Okay, you have to remember the two names. Plant receiving the bud or the shoot is called the stalk and the shoot fixed on it is called the scion. Can you see it is being fixed now? Okay, after that how it will be grafted? For successful graft, it is important that the cambium layer of the stalk and the scion, that means the joining layer, must come into very close contact. Obviously, there shouldn't be any gap between them. so that the growth may continue. The grafted point are then bounded together with a tape. Can you see the tape has been given here also, you can see. And the joint is covered with wax to prevent the dehydration and to protect it from bacterial infection. Please underline it, why it is covered with wax, to prevent it from dehydration, so that it should not get dry, and also protect it from the bacterial infection also. Okay, and slowly you will find after few days the new cell develops and a new plant slowly grows. So grafting also mainly we see few of the plants like rose and all are being done. Now coming to the last process, that is the micro-propagation, because tissue culture is coming under micro-propagation. It is the propagation of plant by tissue culture technique. If vegetative propagation is not possible in a plant, when the other three possible are not taking place, then when we will do with the tissue culture of vegetative buds, how it will be done? When tissue of vegetative buds, shoot, apex or any other suitable part of the plant. Please underline this, that tissue of vegetative buds, shoot, apex or any other suitable part of the plant can be used as a explant for micropropagation. These will be taken for micropropagation, okay, as a explant. Explant means they will be taken for tissue culture. The explant are treated with sterilization chemicals. Sterilization chemical, why? So that no microbial growth will take place, to prevent the microbial growth, and then cultured in a particular nutrient medium. Students, please remember this part, very important, because these are the essential processes should be followed in this tissue culture. The cell grows and divides to form a cell mass called the callus. When the cells start growing inside it, and they form a cell mass known as callus. Some growth regulators, who are the growth regulators? That is the plant hormones, are added to it to give a better result. The callus differentiate into plant parts looking like a tiny plant. Okay, in the long run you see that in the C and D you can find, and here also you can see that they are slowly giving the plant type shape. So small plant, they will be looking like tiny plant, and that's why the name has been given plantlet. Okay, after 4 to 6 weeks the plantlets are transferred into the soil. Then they are taken out from there and transferred into the soil, and there they develop and multiply what? Disease free stalk. In this way they develop. Now this technique also you should know some examples is used to grow what? Orchids, gladiolus. Then this also many are there. In this we can use this tissue culture for this method. Now we are coming to the benefits and limitation of tissue culture. Benefit is what? Rapid propagation of identical individual. Obviously when we are using this same species, then identical, and it is a very quick process also, and this technique is a productive for superior varieties. Remember that superior varieties, it is being very much useful. Now it is very useful in cases where seeds are dormant. Suppose we are not getting the plants with the help of the seeds, at that time we will use this tissue culture and generate the plant as a new tiny plants, and we can use this plant for further growth of the plant. Limitation of tissue culture. It requires a lot of scientific expertise, obviously, because without scientific, these are all scientific things, so it needs expertise in scientific work so that it can be done properly. It cannot be applied to all the cases. All types of plants cannot be used under this, and it is not easily applicable in remote agriculture areas. Since it is a scientific based process, so it cannot be used in remote agriculture areas also. Okay, so please remember these are the limitation of tissue culture. So students, we have learned today the 4 methods of artificial vegetative propagation. Now we will come to the next topic, that is called sexual reproduction in plants. Sexual reproduction in plants, we will see many types of plants bearing flowers. This is the most common features of the sexual reproduction in plants, that these plants bears flowers. and these flowers are born on the shoot part. In the branches you can find that the flowers are growing, and these flowers serves as a reproductive parts of the plant, because they bear the female male reproductive cell, which on fusion they produce seed, and this seed they germinate to give the new plants. So what we understand by this, the flowers are the main reproductive part of this plant. Now the flower is the reproductive part of the plant. To understand how it helps in sexual reproduction, let us see a typical flower and talk about it. Typical flower, you can see here, we have taken an example here also. Floral parts of the most of the plants we will see here. Now first is the stalk. The flower is attached to the shoot. You will see that the flower is getting attached to the shoot of the plant by means of a, what is the name of it? Stalk or pedicel. Okay, the tip of the stalk is enlarged. The tip of the stalk is enlarged and little bit slightly flattened, so that the full flower base will come on it, and flattened from where the petals and the other parts of the flowers arises. This flattened part of the stalk is known as what? Thalamus. So what you understand by the stalk, that it is the attachment between the shoot and the base of the flowers, and then the tip of the stalk is enlarged and flattened from where petals and other parts of the flowers come out, and that part is known as the thalamus. Now coming to the next part, the flower contains main 4 holes. What are the main 4 holes we will see now? First hole is the calyx. Calyx, you can see, it is here also, you can see from the figure that calyx is the outermost part and lowermost also. Outermost, and it is usually green in color. Okay, outermost part of the flowering forming the whole of the tiny green leaves like structure, and these are known as sepals. Okay, in the bud condition they protect the flower. They protect the full inner parts of the flower from the outside atmosphere. In the bud stage you will see that they are fully covered, and sepals are normally what? Green, and it is the outermost part of the flower. Now coming to the next to calyx is the corolla. Corolla is made up of petals. Can you see the colorful petals here? Here also you can see that it forms the second inner holes arranged next to the sepals. Usually petals are white or colored, but rarely green. Okay, they are not found green in structure, but sometimes you find white in color, white petals also, sometimes colorful. Okay, now the petals are normally makes the flower what? Attractive. So petals are large, fragrant. Some flowers are very fragranted also. Some are brightly colored also, so attract the insects. Okay, brightly colored parts of the flower, they are the petals are. Now coming to the next part, that is the androecium. Androecium is also known as the male part. Male part means it helps in the reproduction, which carries the male organ. Now it is the third hole, inner 2 petals. Next to inner 2 petals these are the androecium. Now androecium consists of a delicate thread-like structure. Delicate thread-like structure known as the stamen. Okay, each stamen is formed of a long, narrow, thin filament. Each stamen consists of a long, narrow, thin filament and a broad sac-like bilobed anther. Bilobed means it look like this. That means 2 faces are there. Both are jointed. Bilobed, they are jointed and look like that. Have you seen beans and all? How they look like that. Okay. So bean seed, you will see that they are bilobed. Okay, they are joined together. If you crack them, they will break into 2 halves. So each stamen is formed, a long, narrow, thin filament is there, and a bilobed anther is there. Okay, broad-like structure called anther found at its tip. In the tip, the anther is there, and each anther contains 4 pollen sacs. 4 pollen sacs is there in the anther, which contains what? Pollen grains, from which the pollen grains develops. The pollen grains contain the male gametes. Actually, the pollen grains is they are the container of the male gametes. So stamens, we can say as a whole, stamens are the male reproductive parts of the flower. Now coming to the female reproductive part of the flower, that is gynoecium. It consists of carpels. Carpels, you see in the middle, you see the thin strip is here. Here is the broad and the thin strip, and upper is the small head is there. Okay, the female part consists of carpel. It is known as overall, it is known as the carpel. The fourth innermost part of the flower. It is also called a pistil. Other name of the carpel is a pistil. Each carpel is formed of three parts. Swollen ovary at the base, you can see here also, and a narrow thread-like style. Narrow thread-like style is the just above the ovary. Okay, in the middle, and a terminal expanded stigma on the stalk, and terminal stigma on the stalk. You will see that the head and the style and the ovary. Stigma, style and ovary. These three are the main parts of the pistil, that is the carpel. Now carpel, we can say as a whole, is a female reproductive part of the flower. Now coming to the ovary, what it contains. It contains ovules. Now these are the small rounded bodies present inside the ovary. When you cut the ovary, you can see from here, when you cut the ovary, each ovule contains an egg cell. It contains an egg cell which later develops into a seed. That means ovule in the later develops into a seed after successful fertilization. When the fertilization gets over, the ovule gets matured into seed. Flowers bearing both female and male parts. A flower which contains both the female and male part in one flower is known as bisexual flower. While the flower bearing only male, that is staminate, or female, that is pistillate, are known as unisexual flower. So if you find both the male and the female parts in the flower, in the same flower, you can say that flower as a bisexual flower. But if you find only one of the part, that is the female part or the male part in the flower, you say that flower as a unisexual flowers. Now what is the main function of the flowers? I have already told you that the main function of the flower is the reproductive organ of the plant, because it contains the male part stamen and the female part carpel. The pollen grains from the males, I have told you the pollen sac contains the pollen grains inside them. These pollen grains are being transferred by the air or with the help of insect to the female part of the plant, okay, of the flower. It can be of the same flower also, can be of the same plant, different flower also, or different plant, different flower also. This transfer of pollen grain from anther of the androecium to stigma of the gynoecium is known as what? Pollination. So students, I hope you have understood the sexual reproduction of plant, in which today you have learned the 4 important roles of the flower. So pollination I will teach in the next class. Keep learning, stay healthy, have a good day. Thank you.